Assalamu alaikum wa rahmatullah. Thank you, everyone. Thank you for the organizer committee for their kind invite. It's my great pleasure to present today the genomic landscape in lung cancer. Can you please confirm you are seeing my screen?
Yes, we can see it clearly. Thank you.
Thank you. So the story of lung cancer is an amazing story as an example of a cancer that went through many advancements in a very short time, relatively, from 1997, 13 years ago, with the first EGFR inhibitor in clinical development. The yellow color represent the EGFR story that's unfolded with many biomarkers, with many treatments, targeted therapies. The one in blue, light blue, the ALK and ROS, which started in 2010. And most recently, we start to see more and more large genomic analysis, and the immunotherapy started a few years ago. With an amazing advancement per year. We are seeing every 3 months or 4 months amazing discoveries about either biomarkers or target therapies and immunotherapies. So as of 2015, which is a few years ago, one of the largest studies done in France did the landscape study on lung cancer, especially adenocarcinoma. And as you can see, we have at least 15 different recurrent biomarkers. Most notably is the KRAS, which represent 25%, EGFR and 16%, and other biomarkers with 8 and less. Many of those are target therapies. Others don't have any target therapies like DDR2, NRG1. But the most important ones are usually the EGFR, ALK-ROS1, BRAF, and NTRK. And the problem that we are facing today is simple on paper. We have hundreds of biomarkers. We want to try to link them to approved targeted or immunotherapies. And this is the ongoing process. And we have many guidelines helping us through the process. One of them is the NCCN guidelines. And this is the most recent guidelines that you can see. There's 8 groups. I will not go into any details. My colleagues before me and after me will give some examples. But what we see here is multiple lines of therapies for many of these biomarkers. Some are at the EGFR, ALK, BRAF, ROS, NTRK, MET, and BDL1. So in my talk, I will be more into the detection of the biomarkers. And to understand how we approach this issue, especially when we have hundreds of biomarkers, we have to think about the different molecular levels. Starting at the DNA level, we can detect some of these biomarkers, for example, EGFR, BRAF, and KRAS. At the RNA level, we have the ability to detect ALK, ROS1, and NTRK. We also can detect these biomarkers at the DNA level, but usually is not as good as the RNA. And at the protein level, which is the end product from the DNA, we can detect BDL1 and NTRK using immunohistochemistry. And any changes in these 3 levels use different methods to detect the mutation or the genomic alteration that causes the cancer, and some of these are sensitized by different lines of therapies. And the story a little bit get complicated by the fact that even within a single gene like EGFR, we have at least 500 different mutations. With ALK, we have more than 27 different fusions partners, 12 also for the ROS1. So the landscape is a little, although we have few genes, but each one represents tens or hundreds of different mutation. Each one is detected by different methods. And in the last few years in the lab, we seen different paradigms. Initially, we have a single gene testing, and this is still common, at least in 30% of the labs, where they target each gene, each biomarker by itself, and then they go in a sequential or parallel strategy. Then we start to see small NGS panel, usually 11 to 20 genes. Then medium size NGS panel, 50 genes and above. And lastly, we've seen large NGS panel. And they will talk about the impact of each of these very quickly, and what does that mean to us in the lab, and how fast we can get the result to the patient. So just 3-4 years ago, we used to see single gene testing in a lot of labs, above 90%. And the usual way is to go, for example, with EGFR using qPCR or FISH for the ALK and ROS1 as in parallel, or we go by the most common and going to the least common. So we start with the EGFR since it's appear in 25% of the patients, then to ALK, which appear in 4%, and finally to the ROS, since it's the least common out of these three biomarkers. And the diagnostic yield from this approach from our hospital at National Guard Hospital in Riyadh is we detect 25% of our patients with EGFR mutation, similar to the published literature, 7% ALK, and 1% ROS. So our diagnostic yield is the combination of these. 33% of our patients have a targeted biomarker. The number of reports, however, is 3 to 4 reports between these, and the turnaround time goes from 3 days to 4 weeks, depend if the assays are available all in-house or in send out, and the range between 1,500 to 4,000 riyals. But this is, causing a problem. Not only the turnaround time is long, we have multiple reports. The oncologist need to continuously be in touch with the pathology lab and the molecular lab to find out the results. So it's kind of headache and slow process, and is not cost effective at all when we have 10, 15 different biomarkers, as you saw in the beginning of this lecture. So we changed our gears toward the small NGS panel using next generation sequencing. And we targeted both the DNA and the RNA at the same time. 10 genes at the DNA level, especially the EGFR and the BRAF. And at the RNA level, we had the ALK and ROS and some of the emerging biomarker at that time, RET and MET. So how common is this method or this approach? In 2018, 55% of the labs use next generation sequencing compared to 45 who used single gene testing on the right side. And within 1 year only, there was a jump by 10% adoption to NGS, which increased it to 65%, and less common the single gene. And I think this trend is going to happen in the next 2-3 years, where we no longer will have a single gene approach. It's going to be not cost effective. It will take a lot of time, and more labs will adopt the more complex NGS approach. And today in the markets, we have two major NGS platforms. There are many, 5 or 6, but the most common ones, the clinical grade, are one from Thermo Fisher, and the right side is Illumina. I will not talk about these technical details. I will just highlight how Thermo Fisher technology works. And basically, they have different chips. This is electronic chips. And if we zoom into this electronic chip, you will see thousands of small wells. Under each well, there is an electrode, an ion sensor that will measure the reaction in each compartment. And if we even zoom higher, you will see it has magnetic beads. Each magnetic beads combined with thousands of DNA molecules or RNA molecules that carry one of our hotspots or biomarkers. And the reaction that happens in this well will release different hydrogen ions based on what is the G or A or T or C in the DNA level. Each one will emit a different signal. And the end result is we will read each molecule short segments of the DNA. And the combination of all of this will yield detection of base substitution. For example, you see here KRAS example. This method is very sensitive and very specific, up to 99% for this kind of genomic alteration. If we are talking about small insertion or small deletion parts of the DNA, for example, you see here EGFR exon 19 deletion, you would notice that it's still very good, but less sensitive, 98% sensitivity with this kind of mutation because it's larger and more complex. Another variant or genomic alteration is the copy number alteration. This is basically a huge part of DNA that will get deleted or duplicated. And the sensitivity is a little bit lower. The example here is HER2 amplification. The sensitivity is 95%. And one of the most important alteration are the gene fusion. This is where the DNA breaks between two different parts of the chromosomes and link to create abnormal gene products. For example, ALK fusion or ROS fusions. And this method is extremely good, especially at the RNA level with sensitivity of 99% and above. So this is all great. Then what? Well, once we start having a combined biomarker detection using next generation sequencing, the turnaround time dropped from 6 weeks to less than 10 days, which is amazing given how many biomarkers that we can detect at once. The tissue is not consumed as it used to be because we are now doing all of these biomarker using just one single 15 slides or less. And the number of reports decreased from 4 to 6 to just one report, very comprehensive with a lot of biomarkers at once. So when we did this between 2018 and 19, we added 4% detection for RET fusions, another 4% to detect MET fusions, and 11% for KRAS. So our diagnostic yield jumped from 33% to 52%, even with the smallest NGS panel that we can have, only 11 genes. And the cost is 3,000. So this is fixed 3,000 for more than 11 biomarkers. So what we report in our NGS assay is for every mutation that we detect, we link it to the exact exon level of fusion. Here you see ML4 ALK fusion from exon 13 to exon 20. We supply the allele fraction, basically how strong is the signal. And here we detected this fusion and supported by more than 1,700 NGS reads, which give us the confidence that this is a true positive, it's not a false negative, and no need to confirm this with alternative method. And then we list the approved therapy in adenocarcinoma according to different guidelines. Here you see in green NCCN guidelines, in red the FDA and ASCO in orange and others. And we also list sometimes the therapies associated with resistance for this specific fusion. And sometimes we detect multiple co-mutation in the same assay. And you see here, there is also already ALK mutations, and these are associated with resistance to alectinib and crizotinib mutations. So this helped the physician to guide the selection of the second and third line treatments for this patient. And not only this, we even KRAS is associated with resistant mutation. We start to have like the G12C mutation that is known to be responsive and sensitive for Amgen new treatment. So immediately we went back to all the database that we have for all patients with this mutation and enrolled them into a clinical trial with Amgen. So this is the beauty of testing for biomarkers, even if they are not associated with therapies at that time. Some of the patients may survive for a few years, and by looking back in their data, they could be enrolled in clinical trials as our knowledge and new therapies are appearing in the field. So when we jump to a larger medium NGS panel last year, we added more genes, up to 52 genes, including NTRK. And I think we only got 2 cases so far with NTRK fusion in lung cancer. And we are adding a larger panel, 500 genes or more. And basically what we are trying to look for is if it is negative in all of these biomarkers, we want to see what's the microsatellite status for immunotherapies, the tumor mutation burden, because it's only good to detect tumor mutation burden for immunotherapies when we have at least 400 or 500 genes and above. Smaller gene panel will not be reliable for this. And if all goes negative, we have an arm to send it for research. Basically, we sequence the 20,000 genes using whole exome sequencing and accumulate that knowledge in a database to serve the patients later for any new clinical trials. So The single gene, this is the summary of my talk. The single gene testing used to be great, but it's not cost effective because each gene and biomarker need different methods that require different expertise. With smaller gene panels, we had a fantastic 33% diagnostic yield with adding the BRAF, KRAS, MET, and RET. With the medium-sized panel, we added the NTRK, a slight increase in the number of diagnostic yield, but it's not very great. I think we will see the better diagnostic yield with the 500 genes, but the cost here is almost double or triple the small and medium-sized panel. So it's not always available for all tertiary hospitals. And with that, I would like to thank the Department of Pathology and Oncology Medicine at the National Guard for their continuous help, and to the organizing committee for their kind invite. Thank you very much.